Cell-free fetal DNA analysis. How does the science actually work? This video will explain how cell-free fetal DNA can be analyzed to check for aneuploidies. An example of which is trisomy 21, also known as Down syndrome. So where does cell-free fetal DNA come from? Well, in the pregnant woman, a little bit of fetal DNA that is chopped up starts circulating in the mother's plasma. cffDNA volume varies according to how far along in the pregnancy the woman is and ranges from 3 to 20%, increasing as the pregnancy goes on. There are some samples, however, where you can see up to 50% of the mother's cfDNA is of fetal origin. So once you get the blood sample from the pregnant woman, in order for the fragments to be sequenced, it must be prepared into what's called a library. So how is this done? Firstly, extract DNA using a DNA extraction kit. Secondly, run a PCR. This is to amplify the DNA. Thirdly, you sequence the DNA fragments and align them to a human reference. How would you do this? Well, that original sample that you have is a mix of maternal cfDNA and fetal cffDNA. Each of these strands, no matter what the origin, is a DNA sequence, and the start of each sequence is what you call a read. You can map these reads that are at the start of a DNA sequence by corresponding them to known areas of each chromosome. Here I'll draw out an example of how this is done. So we've already sequenced the DNA and we've aligned it to a human reference. We found out what chromosome it comes from. So you want to draw out or input into a computer the chromosome numbers and how many short sequences, how many reads correspond to each chromosome. So how do we then recognize trisomies, for example, trisomy 21? Say you got 590 reads for chromosome 21 from this sample. Well, you would compare that to a reference, which was a mother and a normal baby. Maternal DNA in a trisomy baby will result in more reads in the chromosome 21 region, for example, than a mother and a normal baby. And this is how you can test to see if there is a trisomy. Scientists would also do a Z-score test to clarify statistically that there is a difference. However, in reality, there's a lot of noise that cancels out that trisomy signal. How would we sort that out? You would filter out the cfDNA that is not useful. Not useful means it maps to more than one region of the genome. Now that we're better at doing this kind of thing, called bias correction, we can be certain of fetal trisomy with only 0.42% of the genome. This was found out in a 2014 study. So in conclusion, this is a massively powerful tool for detection of trisomies.